So if I pull apart this string, then I denature the protein. I come somewhere here. If I release, then I understand it will go back to this. Right? So, studying folding of this protein is not very difficult. And this protein, if you go to a denatured state, it will automatically come back to only this state only. But folding of this, I don't know what will happen. Do I make sense? इसमें सारे चीज है, देखो इसमें थोड़ा हेलिक्स है, थोड़ा शीट भी है, फिर एक लूप है, एक टर्न है, ये सबको फॉर्म करना है। अगर ये प्रोटीन और इसका जो फंक्शन है, which is called ribonuclease, meaning ribonuclease meaning if you give a ribonucleotide, then it will cleave, उसको टुकड़ा कर दो, this protein, that's the function. So naturally इसका जो folding behavior है वो complex है, right? So इधर in folding of ये सारे protein molecule को I would be needing assistance, किसी को help चाहिए। ये थोड़ा ऐसे नहीं होगा, वो हेल्पर है, वो जो प्रोटीन मॉलिक्यूल उसको बोलते हैं chaperones. And this is the best example of a denatured protein. ये जो white वाला है ना, this is actually a cell, अंडा, and ये जो white वाला portion है, ये सब denatured protein, पूरा गया हुआ. When if you have seen a egg yolk, okay. Now I simply show you a few pictures because protein pictures are nice. They are fibrous proteins. You also know, you also have them. I also have them. See this cobweb. They are all fibrous proteins. Okay. This is collagen of leather. See, these are all fibrous proteins. This horn, then this wool, they are all proteins. Protein, keratin. Okay. See all these structures, these are all, how do you see them? Obviously under the microscope. And depending on what you want to see, you design your microscope. If you want to see something very tiny, then you have to go to electron microscope and this is something, this probably I was telling you that not all amino acids we can synthesize, but what we could do, we can store them. So like that egg protein that I saw, mostly, not all, mostly it is ovalbumin, okay? And this storage protein functions in the storage of amino acids. So कैसे होता है? You store the polypeptide and whenever you need those individual amino acids, क्या करता है? उसको एक ACE वाला enzyme आता है, peptidase, proteinase, protease, ऐसा उसका नाम होता है. So वो निकालो, वो जो प्रोटीन है वो आके depending on your need वो आएगा and it will cut into small pieces. And when you cut out polymers, big polymers, you finally end up getting amino acids, right? So this is ovalbumin of egg white. Casein is the क्या बोलते हैं उसको हिंदी में? चना. Chana is the right word. Chena. Okay? So that's the milk if you coagulate milk, right? So that is actually mostly, if not all, is a protein called casein. And that is how these phrase you get them. So this is, these are few proteins that I have already listed, hemoglobin I have talked about. And also, this I did not, but I told you that, insulin, I only did not tell you how it takes part in all these diabetes, but forget that. So these are a few functions of proteins, okay? Antibodies, I don't know whether you have heard of. So, if you go through this table, you will find this collagen, I have already told you, and then in movement, actin and myosin, like I am talking, a lot of my mouth muscles are right, I am raising my hand. These are all functions done by proteins. Only proteins. But the only thing is, there are lots of proteins. It's not only one protein doing the job. That was, that sickle cell disease I showed you, उसमें से एक प्रोटीन का प्रॉब्लम आया और एक डिजीज आ गया. But I give you the best example is cancer. Now, why is people unable to solve cancer problem? Because there are so many protein and gene involvement in it. So, usko deciphering that is becoming extremely difficult. Because it is, in Prabhu's language, it's a multi-functional problem. It's so many variables, So how do you solve an equation if you have, hundreds of variables in one equation? So that is the cancer. And then this is a defense is antibodies, and then catalyzing reactions I have already told you. Now I come to just one more smaller part. How long can I get? Achha, what is this? What is this? Anybody's guess? Anybody's guess? This is actually Mohenjo-daro. Harappa and Mohenjo-daro, those days, right? So that is an aerial view. And where, at least one thing I could see, that this I could see from top, that this could be some kind of a well or a citadel or something, right? But if I come closer, then I see some more structure. So if I am away, I get a rough picture. I don't see much. But more closer I come, and obviously technology has to contribute. So more closer I come, I can even see drainage system. In those days, bathrooms. And these are something that you don't need a historian or some experts to really decipher what are. Just seeing is very important, right? So I will just show you a few things which are very important in biology. May not be only related to proteins, but effectively related to proteins. And these are, please mark the scale here, okay? Scale is 0.4. These are lipid vesicles. But you could see, I showed you a red blood cell picture previously, and you could make out that they look very much closer to a red blood cell. Okay? This is, I just show you the diversity, why is biology so interesting, and why is solving a fundamental biology problem is so difficult. So this is simply the size. They're all different kinds of cells, and I tell you, if there are 1, 2, 3, 4, 5, 6, 7 of them, there is actually 700 of them in our own body. They look different. Okay, if they look so different, then I can simply see them under microscope and tell you this is a megakaryocyte, this is a red blood cell, this is a macrophage. But life is not all that simple, right? There are many cells which look identical, but they are completely different cells. How do I know that? Then there are proteins comes into the picture, and these proteins are basically flags. They act as markers. These proteins, only thing today I will tell you, proteins which are on the surface of a cell, they are very different from all those proteins that I have been talking about since last month. And to give you just one example, 3 such cell surface protein structure has been solved till date. 3 of them. All 3 of them were using the same X-ray diffraction. This is just to show you that, see, these are all red blood cells, but how different they could be when the system is under some kind of a disease. And only thing here I tell you that this disease has nothing to do with red blood cells. It is not sickle cell disease. If it is sickle cell disease, I know that, it is sickle cell disease, so this is RBC specific, so RBC shape is getting changed. But these are some kind of leukemia. Many different varieties of leukemia. But you forget about that. You just see those pictures. Now, this is something I just happens to, this is our work. This is something that happens to be the fact that that is, this is a normal red blood cell membrane, and this is that poor fellow who was suffering from some kind of a very rare kind of a hemolytic anemia. And very interesting, you see, this is his actually pump. He has some skeletal abnormalities. Now, obviously, if you ask me, which you are bound to, are they just fact, or you have found it, or why? There is no answer to why. These are only facts. So more and more such facts are accumulated, more and more you could probably go towards solving, if at all, a particular disease, or at least understanding. And here comes the last part, which is also some kind of 10 minutes I have. So this is, if I take now this word, I am sure you have heard of, it is genome. Because in 2000 in the year 2000, the first draft sequence of human genome was published. So genome is the whole chromosome, because genes are only part of it. There is maximum, actually more than 90% of